Hey everybody, today I want to talk about monohybrid crosses in genetics. Now, monohybrid crosses, as I'm sure you know, are crosses in which you have only alleles at one loci, or one locus, I guess would be the correct way of putting that. Before we get into that, let's go into some people and terms. So, Gregor Mendel, he studied pea plants, and he was able to cross two pea plants to get a different ratio of his crosses, let's say, to put it very simply. He studied alleles, which he didn't know exactly what was going on at that time, but alleles are essentially you get one allele from your mom, one from your dad, and they're a flavor of gene, to put it. So if you think of it as a gene is kind of like a condiment, let's say, and an allele might be mustard and ketchup. You have one of each. That's a very rudimentary way of putting it, but that's a way to think of it. Now, a locus is a spot on a gene, or on a chromosome that a gene resides. I was originally told by my very first biology professor to think of genes and locuses and alleles as the same thing, but once you're in genetics class, you are supposed to go a little bit deeper than that. So we're going to call them three separate things. Make sure you understand the difference between those, because later on they become very important. Now, DNA is what basically makes up genes. And it resides in chromosomes. Chromosomes are what hold our DNA in our cells. That wraps around DNA wraps around histones and holds it in there. But that's not important as of yet. So, to do a monohybrid cross, we have two different alleles. Like I said, or actually, it's going to be easier to draw it. So we have, let's say, big A and little a. Now, this person is going to be a heterozygote. Heterozygote. All that means is that they have one big A and one little a. Now, we can also have a homozygote, like that, and that means they have either two big A's or two little a's. So this person is going to be either big A, or little a, little a. Alternatively, no, we're just going to leave it at that. Heterozygote, homozygote. Now, when we have a cross between these two individuals, let's say, we have one individual that's going to be, let's erase this really quick. There went the camera. That's what I get for buying a cheap tripod. Okay, so we have one individual that we will say is going to be big A, little a. And let's say the other individual is going to be little a, little a. So little a, little a. Now, you see I'm writing them in this pattern. That might seem weird, but there's a method to this. Not the best method. You will actually probably be taught later on to not think of it in this way if you can avoid it and just do simple math. But to showcase it, this is a lot easier. So we're going to draw a square, a very squiggly square, like so. Let's pretend those are even though they clearly aren't. And let's say this is the mom up here. Doesn't matter, this could be the dad as well. But over here we're going to have the father, which I don't know why I didn't just write dad, but same difference. And due to the law of segregation, you are going to get one allele from your mom and one allele from your dad. Now these, remember, I don't know if I ever said that. That could be a problem. These are going to be alleles. And like I said earlier, alleles are flavors of genes. So we're going to get one from mom, one from dad. Hence this diagram we have right here with the 4 different squares. So mom's going to give us either one big A or one little a allele. So we can have big A right there, or little a. And alternatively, we're going to come down here and we have big A and little a. Now, what sorts of alleles can we get from our dad? He clearly doesn't have any big A's over here, so that means he can only give a little a or little a. So, of course, he's going to always give a little a. Now, if we look at the square that we drew, this is called a Punnett square, by the way, we see that we have two individuals that are big A, little a. So we have this one, and we have two individuals that are little a, this one and this one. These are going to be called the genotypes. So I'll write that right over here. So we have genotypes, and like I said, we're going to have the big A, little a, which is going to be half of the progeny. So progeny are your offspring. So let's say you are a male hippopotamus, and you mate with a female hippopotamus. Half of your hippopotamus offspring are going to have the big A, little a genotype, and the other half would be the little a, and that's because of these alleles. Now, if we had two homozygous recessive means a little letter or a lowercase letter. If we had little a times little a, we would get all little a's, which would mean 100% of our genotypes from our offspring would be little a. But in this case, they are 1/2 each. Now, these genotypes are very important. These are what determine everything about us. Little quick fact for you, we as in humans, share 99% of the same DNA with one another. So me and you, the person watching this video, we have 99% of us in common. There's only 1% of us that differs, but every single thing in that 1% is what makes me drastically different than you. Now, two people can have the same phenotype, and phenotype is what these genotypes will determine. So let's say that this big A, little a is a cross. Cross would be big A, little a times little a. Sorry for the handwriting. That would be a cross. So we're crossing the mom and the dad. And let's say that this gene, and remember genes are alleles, so big A, little a is a gene, and the gene consists of two alleles. Usually they will consist of two alleles. Let's say that it's going to account for hair color. Now, most of our genes are determined by two alleles, or have two alleles. Some of them have more than two alleles. Just gonna put that out there, if I remember correctly. Like I said, I am just a student, so anything I get wrong here, feel free to correct me. That would be great. I'll learn something. Hopefully you'll understand it better, and we'll have a good time. So now, like I was saying, we have big A, little a, little a, little a. These are the genotypes. Genotypes. And the phenotypes now are going to be either big A dash or little a, little a. And we will say that the big A gene equals, let's be crazy, let's say that humans are going to have green hair. So big A is going to equal green, little a equals salmon colored hair, because I love the color salmon just because it's such a weird color. Now, this person, because they have one big A allele, that means they're going to have green hair. This dash means that the second one, or actually in this case, it should be a little a. If you see a dash there, that means it could be either a big A or a little a. It doesn't matter. In this case, we knew from the cross that it was big A, little a. So that's why I erased that and put a little a. So this person has one big A and one little a allele. The big A is going to be dominant. Pointing arrow, I'm going to say dominant. And that means that any of these offspring that have the big A, little a genotype is going to be green. So we'll write green right underneath them. Now, we also have little a, and little a, or two little alleles. Again, I don't know if I mentioned this earlier, alleles don't have to be big A or little a. They can be A, B, C, D, E, F, omega. I've never actually seen that. They don't have to be little a or big A or any types of A's is what I was getting at. But these are recessive alleles, And so he's going to be homozygous recessive, or she, it could be male or female. And because they don't have any dominant alleles, their phenotype is going to be salmon colored. It's such a fun word to say, salmon. Now, this is how we determine the phenotypes. So before we said we have one half of the progeny, you're going to have the big A little a genotype, and one half you're going to have the little a phenotype, or genotype, sorry. Now we got one half of them are going to be green because they have the big A little a phenotype, and one half, or because they have the big A phenotype, sorry, and one half are going to be salmon colored because they have the little a phenotype. So there you go. So our answer would be, if we were asked what proportion of the progeny or of the offspring would have the green phenotype, our answer would be 1/2. All right, so if you have any questions, feel free to ask them. I'll probably be going over trihybrid crosses. Hopefully the next video will be not as shaky. And we'll all get a better understanding of genetics. Now, go out, have some fun with it, study it, work a few problems, and see where that gets you. All right, take it easy.